pivoting towards nucleotides. So we're going to be talking about anabolic, catabolic, and salvage pathway as well. Just quick refresher on nucleotides. We have deoxy versus oxy. DNA does not have an OH there on the two. Oxy for RNA does have an OH on the two. C's and G's are going to have 3 hydrogen bonds in a double strand. A's and T's are going to have just 2. I always remember that C is the third letter of the alphabet. 5' to 3'. So if we just look at this 1, 2, 3, 4 tetranucleotide sequence, the carbon over here, carbon number 5, right? 1, 2, 3, 4, 5. Carbon number 5 is not attached to another nucleotide. So this is the 5' end. And if we go over the other side, well, carbon number 5 is attached to something. Okay, well, that's not interesting. So then it must be carbon number 3, because that's not attached to anything. So I know that this is the 3' end. Pure as gold, purines, adenine, guanine. We all know that. We've heard that. But I would remember that it's pure as gold ingots. Ingots shown over here because inosine is going to be an important purine precursor as well that you might not have heard as much about before. And then cut the pie, pyrimidines are cytosine, uracil, and thymine. Of course, uracil is only an RNA, and thymine is going to be DNA. I would know the shape. Pyrimidine is this, while purine is this. I don't know why in my head, but I always thought like, you could stuff a lot of pyramids in the pyrimidine a little bit more. And then I also thought, like, I guess if the pyrimidine was a shape, you could build a pyramid out of that shape. So if you want to think about pyramids fitting in a pyrimidine or pyrimidines building a pyramid, more so than purines, for whatever weird reason, that worked for me. Maybe that's me specific, though. All right, anabolic. Let's build some purines. What's going on? So we got glucose, pentose phosphate pathway. This gives us ribulose 5-phosphate, which can isomerize to ribose 5-phosphate, shown yeah. We then can PRPP, phosphoribosylpyrophosphate synthetase, bunch of fancy stuff just to say we put a pyrophosphate right here on carbon number 1. We've now made the ring. We have made the ring, and I guess put the phosphate on as well. Now, the first step after this, amidophosphoribosyl transferase. This is the rate limiting step. Really, all this does is swap this with a nitrogen group. Probably not even that important to know what that is. Maybe I would know that this is the rate limiting step. And then we're doing a whole bunch of other steps. Not important to know. Know that we're putting in things like glycine, glutamine, formate, aspartate, and you can actually kind of trace these to see where they end up in the purine. At the end of all of this, we're going to have inosine monophosphate. Now, keep in mind, we are talking about the production of RNA for all of this, as we can see with this 2' carbon. Pure as gold ingots here, inosine. Inosine, so we now made the nitrogen base on top of the ring, right? Here was our ring, we made that first, and then here was our nitrogen base, we made that second. We can now make this into AMP or GMP. That's how you make a purine. Negative feedback as expected. You got enough GMP, it's going to go negatively feedback. Got enough AMP, negatively feedback. Got enough INP, negative feedback, right? Just you should have an intuition about what the feedback should be like. Pyrimidines. Same making the ring step, right? Ribulose 5-phosphate becomes PRPP. So just memorize PRPP as the base nucleotide, the base ring. The base ring. No, I should, as just the ribose sugar, right? So when I say base, I'm saying as like the fundamental unit, not to be confused with the nitrogen base. So I misspoke. It's confusing. Okay, so here we made our simple ring, which we want to attach our nitrogen base to. Previously, we did all of this, and then we did the nitrogen base. Second, these are kind of happening like in parallel. So now we're making the nitrogen base. Ammonium and carbon dioxide. That sounds familiar. That's the beginning of the urea cycle, if you think about it. Ammonium and carbon dioxide, and that makes carbamoyl phosphate. But remember, that was carbamoyl phosphate synthetase 1. This is synthetase 2. And if you remember, the first 2 steps of the urea cycle happen in the mitochondria. This happens in the cytosol. They love to ask little shitty questions like that. So I would just know that this is all in the cytosol. In the urea cycle, it was a long track of in the muscle would break down, make a nitrogen, put it in a glutamate, put it in alanine, ship it through the blood, get it to the liver, we convert the alanine back to a glutamate and rip off the nitrogen from the glutamate. That's where it is. Here, it actually comes from a glutamine. Anyways, we made our carbamoyl phosphate. Great. Carbamoyl phosphate then combined with ornithine in the urea cycle via the enzyme ornithine transcarbamylase. Well, in this case, it's going to be carbamoyl phosphate that combines with aspartate. And similarly, it's, in the urea cycle, we had carbamoyl phosphate and ornithine. And the enzyme is named after ornithine transcarbamylase. This one is kind of similar in that it's not named after carbamoyl phosphate. It's named after the other thing, the aspartate, or the other thing, which was previously ornithine and is now aspartate. This is aspartate transcarbamylase. Also recall, anachronistically, I mentioned earlier that if you have an ornithine transcarbamylase enzyme deficiency, a genetic deficiency, you will have a buildup in orotic acid. Now we know why. Because in the urea cycle, you would get a lot of this and you couldn't really do anything with it. Well, that was for the urea cycle. It's going to escape the mitochondria, make its way to the cytosol where it will react with aspartate transcarbamylase and make a lot of orotic acid. So the orotic acid buildup hopefully makes sense now. We've now made the nitrogen base, we made the ring, but now we got to slap them together. So UMP synthetase is going to combine these two, and then from there, we can make it into cytosine and thymine. And again, we have been dealing with these ribose structures. That's when we're doing pyrimidine and purine anabolic processes, we're making RNA. So if we're making a thymine, that's just by definition DNA. So you need to do ribonucleotide reductase, which, if we're going to reduce something, we're going to get rid of an oxygen bond and or gain a hydrogen bond. And that's exactly what happens, right? This becomes a hydrogen. Well, that's how we get it from RNA to DNA. What about, the fact that uracil and thymine have different nitrogen bases? Well, we're just going to add a methyl group via thymidylate synthetase. And remember that this uses, from vitamin B9, vitamin B12, all of that fun pathway, 5,10 methylene THF is going to add that methyl there. Negative feedback as expected. Bam, 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 right? The downstream things inhibit the previous steps. We saw this for uracil making thymine, but it's going to happen for any time, we want to make an RNA nucleotide into a DNA nucleotide. We have this, and then we reduce it to become we add a hydride. Negative feedback as expected, right? If you have enough DNTP, it's going to come back and inhibit this. So ribose 5-phosphate becomes PRPP, and this does a whole bunch of steps to make IMP, which goes here or here. Well, if you have 6, a lot of these are chemotherapy drugs. Some of them are immunosuppressive drugs. 6-mercaptopurine. Purine azathioprine. Technically azathioprine, I think, just becomes 6-mercaptopurine. But I would just note the purine. So this is going to be a competitive inhibitor for somewhere in this process. Important to know. If we go the other route, remember we had glutamine, which had the nitrogen and CO2. CPS2 makes carbamoyl phosphate combined with aspartate via aspartate transcarbamylase. We make orotic acid. We now combine that with the PRPP to make the uracil. Ultimately, that makes cytosine. And, we want to make a thymine, but remember, we got to do ribonucleotide reductase. Well, hydroxyurea, we're kind of random. This will inhibit ribonucleotide reductase. And just let's make super clear, it's not just inhibiting this one instance. We're going to say it's inhibiting everywhere. Anytime you want to take an RNA to a DNA, hydroxyurea is going to inhibit that. Also, hydroxyurea, just random note, we saw it as a treatment for sickle cell, and that is going to induce fetal hemoglobin production. 5-fluorouracil, again, you should just think about the name. The name uracil, is it going to affect anywhere on the purine side? No, it's going to specifically affect the pyrimidine side. It's probably going to act as a competitive inhibitor. It competes and inhibits thymidylate synthase, which again is interacting with 5,10 methylene THF. Methotrexate, we said, is a competitive inhibitor here in TMP. which is component of Bactrim, is going to also inhibit this. Breaking things down. Pyrimidines are simple and boring and probably not going to get that many questions. These puppies break down to malonyl-CoA. This breaks down to methylmalonyl-CoA. This should make sense. What's the difference between a uracil and a thymine? A thymine has an extra methyl group. So this is 3 carbons, and this is 3 carbons plus this carbon over there. But this is the low yield. Much higher yield is this. We did pyrimidine catabolism. Now let's look at purine catabolism. So all of these are nucleotides. IMP normally becomes AMP and GMP. That's not a breakdown. Now the breakdown is that IMP, AMP, and GMP can all lose their phosphate. If you lose your phosphate, there's 3 things for a nucleotide. You got the phosphate, you got the ribose, and then or the deoxyribose or whatever. I'm just going to use them interchangeably for the most part, unless I'm making a very specific point. So the phosphate, the ribose, and the nitrogen base. First, we're going to take out the phosphate. Now it's a nucleoside: adenosine, inosine, guanosine. Adenosine, and you should know this, and it's going to come up later in more genetic stuff, is that adenosine can be oxidized to inosine. So this is adenosine. Maybe not oxidized. It can be converted to inosine. Adenosine deaminase. We can then rip off the ribose sugar, or the deoxyribose, whatever. And now we're making we just have the nitrogen base. We have an adenine, hypoxanthine, and guanine. Adenine ADA can turn into hypoxanthine. We can now break these down. Guanine can be broken down into xanthine. Hypoxanthine can also be broken down into xanthine via the enzyme xanthine oxidase and guanine deaminase, respectively. Xanthine oxidase then works, and this is where it gets its namesake, it then works on xanthine. It is going to break down xanthine, it's going to oxidize it into uric acid, which is very important for a couple diseases we'll talk about in a second. This can theoretically be converted to allantoin, which is a bit more water soluble, and some medications try to exploit that. But this is the entire breakdown. This is how we break things down, but we want to be very green, right? We want to recycle as much as we can. We want to make cuts and be effective where possible. So there's what we call a salvage pathway, where we can, as we're breaking stuff down, our body goes, you know what? Maybe we shouldn't throw this out. Maybe we can actually put this back in the system rather than making another one from start. So these are just the nitrogen bases. Remember that PRPP is just the ribose unit, the fundamental unit, with the phosphate. So if you could just slap them together, you can just get back to your nucleotide again. And that's exactly what this enzyme does. Hypoxanthine guanine phosphorybyl transferase. So phosphorybyl transferase means transferring, adding it to. And hypoxanthine and guanine, well, notice it only works on hypoxanthine and it only works on guanine. It takes a guanine and combines it with a PRPP, and boom, booyah, you got a normal looking G nucleotide. Hypoxanthine can make an I nucleotide, which can then, of course, just become an AMP and GMP. So wait, does that mean that it's the name is very specific: hypoxanthine, guanosine, phosphoribosyl transferase. It only works on these two. Does not work on adenine. Does that mean adenine can't be salvaged? No, because remember, adenine, adenosine, doesn't matter. Both of these can be converted to this middle pathway. And if we make it to inosine or hypoxanthine, that can then just go up ahead and again be salvaged. So it's indirectly salvaged by being converted to inosine or hypoxanthine. So that to me that's a lot. Like, I do think this is stuff you should know, but you want to simplify it. This is probably going to get you majority of questions. Purines are broken down via a lot of random ass processes to hypoxanthine. Keep in mind that you do need ADA technically to break down the adenosine and the adenine, because you got to convert it to inosine or hypoxanthine. But there's a bunch of things to break it down. So purines become hypoxanthine, and then xanthine via xanthine oxidase becomes uric acid. That is the catabolic side. The salvage side is, you know what? Maybe we could just recycle this nitrogen base by adding on a PRPP. And so hypoxanthine guanine phosphoribosyl transferases are going to combine, PRPP plus our nitrogen base, and boom, we got a nucleotide. So this is how the salvage is working. So when we're breaking things down, a couple deficiencies that you could have, an ADA deficiency, you can't break down your adenosine, it's going to accumulate and it's going to cause SCID, which we'll talk about in a second. Xanthine oxidase, this is important. It is a target for people with gout. We use allopurinol or febuxostat, which I think is less common for some reason. And on the salvage side, again, ADA could also be considered an a catabolic issue and also a salvage issue because, you know, it's kind of you've got to catabolize something to salvage it to a certain point, and ADA doesn't let you catabolize it. So if you can't catabolize, you can't salvage it. So that's why we see this show up on both sides. But it's important, you need to know it, but I don't want to inflate the importance. So I'm talking about it a lot because I personally found it confusing. So ADA deficiency could theoretically be catabolic or salvage. More importantly is this one, and this one, in my opinion, HGPRT could cause gout, but more importantly, Lesch-Nyhan syndrome. So SCID, severe combined immunodeficiency syndrome. In other words, your T cells suck. There's a lot of different ways we can cause this. The main one is that 46% of cases, the gamma chain on your T cells doesn't respond to cytokines, and this is X-linked. But you know what? The one that probably is relevant is going to be the one with the enzyme of nucleotide metabolism. So an ADA deficiency. This is autosomal recessive, and you can't break down the adenosines. So, you know, if we break down, if we get rid of those enzymes, this is just going to become some sort of toxic, random stuff. And this is bad for the lymph nodes, and it causes the T cells to die. But also, you can't salvage, and if you can't salvage, you probably can't make enough nucleotides to keep up with the replication rate. So this could also affect like T cell production in the bone marrow. And you know, the Boy in the Bubble and silly Jake Gyllenhaal movie is based on this. Gout, called the disease of kings. We'll see why. It's like a type of arthritis due to, like, if you have too much uric acid in the blood, it becomes something called monosodium urate, and it starts, for whatever reason, clumping together, and then it deposits in joints, and it causes an immune response, which just generates a lot of pain. So where does it accumulate? Podagra is the fact that it typically is going to accumulate and cause a lot of pain on the MTP joint of the big toe. It's probably going to be asymmetric. You know, it's going to affect, because we're just talking about like these uric acid things floating around in the blood. And so it's like, it will happen on both sides, but it'd be weird if it was exactly equal deposition on both sides. So that might be a way to distinguish this from like just arthritis. Also, you know, if it goes to the big toe, you might think gout. You're also going to get what's called a tophus, where it's depositing in the skin, and you can see right there to develop. 90% of the causes is actually just the fact that you have an issue with your kidneys, or you're on diuretics, and so your body can't get rid of the uric acid the way that it normally does in the urine, and so it's just going to build up, and it groups together, clumps together, deposits in joints, causes a lot of pain. Could be the case though that you're overproducing. Less common, it could be the case. So HGPRT deficiency. So let me think. Yeah, so typically we want to recycle hypoxanthine and push it upwards in the cycle, right? To remake our nucleotides. If that's the case, well, then I'm not going to break down as much hypoxanthine to xanthine to uric acid. So the salvaging is good because it's good to recycle, but it also prevents us creating this toxic uric acid. So if you have an issue there, you're going to have more uric acid. Von Gehrig's disease, you can't do glucose 6 phosphatase. So you can't push glucose out of your blood in your liver. And remember that glucose can go make glycogen, it can make acetyl-CoA, it can be pushed out of the blood, which we can't now, and it can also do the pentose phosphate pathway. So if you can't push it out, then you got more glucose, and some of it's going to be shunted in the pentose phosphate pathway, which means we're going to have, and one of the things that PPP does is that it makes the ribose sugar unit, and so we're going to have more of this. You might have more nucleotides and therefore more breakdown and more uric acid. Tumor lysis syndrome. If you have a tumor, cells are dividing a lot. There's a lot of nucleotide generation. You're making a lot of nucleotides. You're breaking down. You have tons of uric acid in your cancer. And you know what? Those cells are shitty, and they're dying, and they're spilling uric acid in the blood. And it's called tumor lysis syndrome. This is one of the many features of tumor lysis syndrome. But spilling the nucleotides and the uric acid into the blood this can cause gout. Additionally, now the surgery, the trauma, you get into a car accident, you have surgery. It's not necessarily like the cancer, you're making a lot of nucleotides, but it is the second part where if you're damaging a bunch of cells, they're going to spill a bunch of nucleotides out in the blood. That can cause too much uric acid, which can cause gout. Now, that's like those are the underlying causes we just talked about. But it can be exact. So someone might have a gout and then just not have an attack for a couple of weeks, or they're fine, and then suddenly they have a big attack. What might precipitate it? What might exacerbate it? Meat and fish and alcohol, and that's something that like wealthy people used to eat. So this disease has obviously been around for a long time, and they just noticed that wealthy people got it because they're eating all of these rich foods and drinking a lot. That's why it's called the disease of kings. Meat and fish, a lot of nucleotides, a lot of uric acid. Boom. Alcohol, dehydration. If you dehydrate, you're gonna have less fluid in your blood, which means higher however much uric acid you have, the concentration is now higher. And if the concentration higher, it's more likely to precipitate and deposit. Also, alcohol is going to compete for the same transporter that is going to help secrete uric acid into the urine. Treatments, allopurinol and febuxostat. So how are these working? Hypoxanthine is made into xanthine. Xanthine is made into uric acid. Same enzyme twice, xanthine oxidase. Well, if you inhibit it with either of these two things, you can't make uric acid. Hypoxanthine levels will accumulate. Maybe you'll have more salvage. Maybe this is discarded some other way. Maybe it's just less toxic. Who cares? But the point is we're not making uric acid. So hooray, this will help. Remember that part of the pain is because, you're just depositing a lot of these monosodium urate crystals into your joints. But then the immune system, kind of like with the macrophages and the phone cells, is trying to help, but it's just doing a shitty job. It's just making things worse. So you actually want to inhibit the immune system, and that actually might cause less pain. So that's where NSAIDs and corticosteroids, and we saw this is inhibiting the arachidonic pathway, right? And colchicine, which is inhibiting microtubule polymerization, which is necessary for neutrophil chemotaxis. All of this is just trying to disable the immune response so that we have a little bit less pain. Uric acid levels should be higher. I hope that one makes sense. Notice, though, that theoretically you could have normal sodium and plasma levels. Actually, you know what? Now that I'm thinking about it, theoretically, I guess your urinary uric acid levels could be higher or lower. If you're under-excreting, theoretically, your urine acid levels could be higher. So mostly what we're looking here for the lab findings is the plasma uric acid levels. So they might be higher, and that should make sense. But what's interesting is they might actually be normal. For reasons I don't really know. They might be normal. So someone comes in, so this could be on your test. They go, but the person's uric acid. It's a trick. It's a trick question because theoretically the uric acid levels could look normal. I mean, it's for whatever reason there's still too much, and there's still the deposition of the crystals and whatnot. Also, you'll see yellow-blue like needle-shaped crystals that have a negative bifringence, which is this test they do where the refractive index of something is going to vary depending on the polarization of light. I don't think you don't need to understand like the physics of it too much. Just yellow-blue, needle-shaped crystals, negative bifringence. Lesch-Nyhan syndrome, so an HGPRT deficiency, so we see this right here. So if this is inhibited, you know what, this actually should be over here. It's in the wrong spot, HGPRT. Boom, it's right there. So it's part of the salvage, right? So purines are broken down to hypoxanthine. Xanthine is broken down into xanthine, which is then broken down into uric acid. Uric acid can be recycled via HGPRT. If we do not have this, you cannot recycle, which means you have more uric acid. Well, again, this is one of the things that can cause gout. But then it also causes this whole other slew of things in this particular case, which is the Lesch-Nyhan syndrome. So more uric acid. Here you will definitely have more uric acid in the urine. And again, I'm glad I made that clarification earlier because remember the other case, most causes of gout, 90% of them are because your kidneys aren't working. You can't properly pee urine out. So the uric acid levels could be low, could be high in the urine. Here they're going to be high because your kidneys work fine. It's just the fact that you can't salvage, which means you're creating extra uric acid. And these crystals, it looks like sand in the urine, and then there could be blood because it's damaging the endothelial layers. So that's one aspect. Another aspect then is that, you're probably going to ramp up more de novo synthesis because you're not salvaging, but it just might not be able to, like, I don't know the rates, but like maybe it's not so easy just to ramp up the de novo pathway and completely compensate for the lack of a salvage pathway. It might be the case that even though you ramp this up, you still just have less nucleotides, which means you're not developing properly. You need a lot of cell division to grow and mature, and it's just not enough to for embryogenesis and just general development to occur normally. It's X-linked. X-linked typically means mostly boys, some girls. I think this would be all boys because I assume that there's very little reproduction, if any, for someone afflicted with Lesch-Nyhan syndrome. It's quite severe condition. Intellectual disability, very, 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 very interesting and unique and horrifying clinical finding is the it's hyperaggression combined with the intellectual disability and maybe some other stuff. That there's self-mutilation. Often they have to have their arms pinned down. Sometimes they have to have some sort of mouth guard or something to prevent them. They'll bite their lips off and they will try to bite their fingers. They'll bite their fingers off. I mean, it's absolutely horrible. They also have dystonia, and so they're just going to have sporadic muscle spasms. Whew, okay, that was it. Great. I really hope I got this out to you guys in time. If not, I'm sorry. I'll try to get the next one out sooner. I hope everyone's doing really well and kind of getting off to a good start to med school and trying to find their pace and everything. So all the best, guys and gals. Sorry.